Get started then. So what we want to refer to tonight is the seminar of the academic year. And so we're of course delighted today to have Dr. Nicholas Trahearn join us in the today. And Nick's one of the particular TI centre alumni, so we expect to be back in 7 or 8 years. So Nick is a postdoctoral training fellow at the Institute of Cancer Research, began his work in district pathology as a member of the TI Centre from 2013 to 2017, where he was awarded a PhD for his work on serial section registration and multi-IHC analysis. In 2017, he joined the Centre of Evolution of Cancer at the ICR Institute of London. Using image analysis and AI from this, his research during this time was focused on the question of how aspects of the tumor microenvironment influence tumor evolution and patient outcome. In particular, he's interested in finding relationships between these microenvironmental features and other forms of biological data, such as genomics and effects of immunoproteins. And one sort of things for today is investigating spatial diversity of prostate cancer microenvironments using AI. Thanks again for joining us, and you were
Oh, thank you, and thank you all for coming. And I think on a personal level, it is really lovely to be here today after so many years away. It's wonderful to see how the group has grown in that time, and really had lovely conversations with many of you here, learning about what things you're working on. And yeah, it's incredible to see how things have developed in the time I've been away. And so yeah, I'm here to talk about some of the things I've been working on since I left Warwick, particularly my time at ICR. Just a sort of update, I have recently moved to UCL. However, for the sake of keeping all my slides the same, I've prepared this a little bit ago. We'll stick to the ICR branding, but yeah, this is some of the work that I have been doing during that time, and particularly investigating spatial diversity. And I think diversity is one the really key word, and how the interplay between diversity and evolution, and understanding prostate cancer and how that affects outcome. So just as just a bit of a heads up there, if you haven't already seen, so most of what I'm going to be presenting today is taken from this paper we had out in Nature Cancer earlier this year. So if you want to take a look at that, here it is, QR code over there. And yeah, I'm going to be talking you through some of the, particularly on the digital pathology side of things, but we'll also dip into some of the genomics and how the two interplay with each other. So just a bit of background, prostate cancer is a huge contribution to the cancer burden, particularly in the UK, but worldwide. So you're looking at over 50,000 new cases every year and around 12,000 deaths each year. And this is the most common type of cancer in males, and the second most common cause of deaths. And I think there are specific things about prostate cancer that make it particularly challenging to work with. One of the things that is particularly I found is the very long time spans in which you can encounter progression, encounter disease recurrence. And this is on top of the fact that it's already a disease that presents quite late into life. And so therefore, trying to sort of understand how this affects things like survival can be quite challenging. And so I think one of the key things is that you need long-term clinical follow-up. Just a few years, which might be sufficient for certain cancer types, is not really sufficient in prostate cancer, because you aren't really capturing the full risk profile, which can go on for many, many years, into decades. And I think that particularly for patients with locally advanced prostate cancer, so patients who have not developed a metastasis, but have high risk features, that would suggest that they may go on to develop metastatic prostate cancer, are a particularly difficult group, often having around a 50-50 chance of recurring or not. So therefore, trying to understand specifically what causes that particular 50% to go on to progress, or patients who don't go on to progress, is an unsolved problem, and further risk stratification is needed beyond the existing risk stratifiers. So a lot of what I'm going to be, all what I'm going to be talking about today is based around the IMRT clinical trial. And this is a phase 1 clinical trial, quite a long time ago now, conducted at the Royal Marsden Hospital. So you can see patients were enrolled between the years 2000 and 2013. And so in some ways, treatment has moved on. But I think some of the specifics of this cohort mean that it's a unique opportunity. You wouldn't be able to collect these sort of data that, using modern treatments. And so these were all patients diagnosed with locally advanced prostate cancer. So these would be patients who, on a TNM staging, they would be M0, but they may have a node status. T stages you'll see can vary from 1 to 4. These are patients who were evaluated as high risk using the criteria at the time. So these would be high PSA, high T stage, or high Gleason grade. And these are patients who were treated with androgen deprivation therapy, and IMRT stands for intensity modulated radiotherapy. So essentially, what the key to intensity modulated radiotherapy is that rather than having, say, a single beam of radiotherapy, you have essentially a specifically shaped beam, which can potentially have different levels of intensity at different parts. So you can really target the area. One of the challenges, particularly in the case of prostate cancer, is that, well, with any radiotherapy really, is that you can potentially cause damage to surrounding tissue, what you don't want to, and particularly in the case of prostate cancer where you have the bowel nearby, there is a concern about, having too high of a dosage. And so therefore, previous to the IMRT trial, there was often lower dosages than would, perhaps be optimal if you wanted to really eradicate the cancer. And so this was the key of the IMRT trial. First off, can you apply a more sort of like a precise boundary around the area in question that you want to target? And can you dose escalate? Now you've got a more precise area, can you hit the cancer with a higher dose if necessary? And first off, does that happen? And so this was a phase 1 trial. So it was not so much about showing efficacy, but rather showing that this was safe. And there's been subsequent trials, the CHIP trial, which is attempting to show that this is a superior way to treat prostate cancer. And so, yeah, a total there was 471 patients enrolled, although we worked on a smaller subset based on tissue availability. And I think what's key, and this is what really attracts us, this is the 12 years median follow-up. And so it represents a very unique cohort, a cohort with very specific treatment, risk of specific treatment, and also long-term follow-up. And just to quickly jump in, so androgen deprivation therapy is used in conjunction. So this essentially suppresses the patient's testosterone to a very low level, as close to zero as you can. And therefore, this sort of is intended to slow the disease. It won't eradicate the disease, but it slows the disease in its tracks. And so therefore, combining this with IMRT, you can slow the disease in its tracks and try to eliminate as much as possible by using intensity modulated radiotherapy, both on the prostate and also the pelvic lymph nodes. So that's a bit of background. And just to give you an idea of the data we're working with, this is predominantly FFPE samples. And the key is that they are taken at diagnosis. And so here we have an opportunity to predict what is going to happen down the line based, and again, you're talking about 12 years of follow-up, so potentially a decade of disease progression, what's happening during that time. And so this was mainly TRUS transrectal ultrasound biopsies. Treatment now is slightly different in terms of sampling, but this was what was available at the time. So you're looking at perhaps around 12 biopsy cores, you can see there, sampled across the prostate using ultrasounds as a way of guiding. And I think one of the key challenges was the fact these FFP samples, which for histopathology is great. This is gold standard. If you've ever seen frozen sections, the tissue quality is much worse, and so this is really the way. But certainly for sequencing, it's the opposite. And so we had a lot of challenges in terms of sample preparation in order to get a good like-for-like comparison with the DNA sequencing versus what we see in the tissue. And from this year, we generated a multi-omic data set, H&E slides, and we had low pass whole genome sequencing. And so this is a low resolution view of the genome. And this again, relates to the fact that we're working with FFPE samples. So we do have deep targeted sequencing for a few key driver genes that are known to be related to prostate cancer. And so, yeah, this was a fairly unique combination, matched histology, sequencing, and also long-term clinical follow-up information for a sizable subset of the patients. And so, yeah, we can produce copy number profiles and also look at the tissue and see how they relate to each other on the same sample. So you can see here, these are copy number profiles that were taken, and this is the analysis, digital pathology analysis, doing Gleason grading on a sort of per gland segmentation level. And from that, you can generate phylogenies. You can see what is the interplay between particular mutations and the Gleason pattern. And so, yeah, this was a unique opportunity to be able to combine those two together that the IMRT trial really allowed us to explore. And so just to give you an idea, yes? Oh, yeah, go ahead. Do you want me to go back? So it's not either or, it is that they will be given the two together. So the idea is one slows it down, the other eradicates, yeah. So basically, the radiotherapy course was so there was basically 5 trial arms. There were different strategies, and so some of the arms were added in later. So like the early arms were simply different levels of radiotherapy, but then they also looked at hyperfractionation. So if we escalate over time, does that help? So they were sort of so. For the most part, yes, it was all IMRT all the way through, but there were also different arms in terms of how it was treated, basically. I suppose so. Yeah, there were multiple different centres. Yes, so there probably would be some variability there. I'm not so clear on the yes. Oh yes. So this was driven like it's driven by ultrasound. So the ultrasound would be used to try and identify where the lesion was, and then you put in the needle. I mean, this is not 100% accurate, and sometimes you'll see there is a certain portion of normal samples we have within the prostate. So let's say the way in which sampling tissue is taken now has changed slightly, but this was what was standard of care at the time. For all the patients, yeah, it was all transrectal. So, well, the majority are transrectal sound biopsies. There's a few TERT biopsies, which is a transurethral resection of prostate, which is a very complex process I won't explain here, but basically you're looking at resected sections, so larger chunks of tissue, but for the majority we're working with sort of needle core biopsies, taken with that way. Oh, yes. I believe it was, I don't remember the exact number, but it was something in the low double digits. I will show a heat map later on so you can see all the key genes there. And one other thing to mention while we're talking about differences in treatment with the ADT, so there are numerous different ways in which you can suppress testosterone. So you have like the LHRH agonists, you have antagonists, which essentially shut down the production of testosterone, but you also have others which stop the metabolization of testosterone. So there is a little bit of variability, but the majority are on either LHRH agonists or antagonists. So, you will see, and we'll talk about testosterone a bit later on, but yeah, testosterone levels just drop to 0 in those cases. Well, close to 0, yes. So the goal of the trial was, so it's a phase 1 trial. So it was trying to show safety of IMRT, not necessarily even that it was superior to previous radiotherapy, just to show that, having this sort of like IMRT radiotherapy and also dose escalation, these were safe. They didn't lead to additional danger to the patient, and in terms of like toxicity, damage to the bowel, for instance. But then obviously there is a phase 2 trial and phase 3 trial, which is involving a different cohort called CHIP, which has been going on since the completion of IMRT. Oh, I'm sorry. Oh yeah, go ahead. Oh yeah, go ahead. Yeah.
About that.
Can you hear Nick speak in just a couple of moments?
Would you like to say something just so we can get an idea?
Perfect. Yep, all good.
Oh, fantastic. Well, apologies for that. Hopefully, if you need me to repeat anything or if there's any questions online, please do go ahead. Well, I can't remember what I said, so that'll be a challenge. But were there any other questions in the room or online before I continue? Okay, well, one thing to say is so that we started, 471 patients were enrolled on the trial, but due to availability of the material, some of them, they've been depleted, and some that wasn't available, there were a few people who had requested that material to be returned. But we ended up with 250 patients, which we had sample. And from those, 100% of the patients were imaged, so we had H&E and produced 1,923 core biopsy whole slide images. But then from that, there was a selection process by which ones went for sequencing. Again, due to quality issues, we needed to ensure that there was high purity, high purity, lots, the portion of normal and therefore noise would be kept relatively low. And so we selected blocks that were 70% pure according to pathological assessment. And so we ended up with 111 patients and 578 core biopsies from the original set. And due to the selection criteria, of course, you select for higher purity, you also somewhat select for higher risk. And so the risk profile of the two cohorts does differ a little bit, but you can see, I think they are relatively comparable. So you can see that generally IMRT works quite well. The proportion of patients who are alive after, the 12 years of follow-up, or median 12 years of follow-up, is about somewhere between 2/3 to 3/4. And the majority go without a recurrence, but you can see that of the ones who do recur, a large portion do eventually develop a metastasis. And so being able to identify which patients are at risk is quite important for that reason. And you can see that these are high-risk patients. So I don't know if you're familiar with PSA, prostate-specific antigen. This is the standard blood test that a patient will have. And so when patients were enrolled on the trial, they would basically undergo radiotherapy at the outset. They would be given ADT to reduce their testosterone for 1 to 3 years. And then they would be checked for roughly every 6 months for their PSA levels. And if their PSA levels start to rise, that's an indication of potential recurrence. And then they may be subjected to additional testing to check for metastasis. And so generally a high PSA would be greater than 10, although we looked at greater than 20 because we wanted to sort of get an even split. But you can see the vast majority of patients have very high PSA and are in the highest grade. So this is while not metastatic, they are a high risk group who have a greater suspicion for metastasis if they go on to recur. So looking at the overall data, we have a few different, like I say, over 12 years collected longitudinal data, and we have overall survival, though overall survival is challenging due to the fact these are older patients, and often death is not necessarily death by cancer, and so therefore it is a bit noisy. So we tended to focus on recurrence rates and metastases. We also have looked a bit more recently at treatment resistance. So if a patient goes on to a different form of ADT or chemotherapy or something like that. And we have a few clinical parameters. So of course, PSA very important. This is the way that recurrence is first detected. But we also have TNM staging. This is at diagnosis. So they may develop a metastasis later on, but this is the TNM stage at diagnosis. And also Gleason grading. Now, one thing to mention about Gleason grading is that every patient who was enrolled on the trial would have been graded by the original pathologist who looked at their case. But the problem is that occurred over a decade. And right in the middle of that, in 2005, there was a redefinition of the Gleason grading system at all. So it is a difficult data set to work with. And so as part of this forecast project, we had a central review pathologist, the Royal Marsden Hospital, review everything by modern, modern, the 2016 revision. So as modern as, there hasn't been a major revision since then. And this review pathology also included a few additional features, such as invasive ductal pattern, which is sort of not part of the main grading system, but is a sort of pathology which carries a higher risk, and also small cell pattern, which can be indicative of neuroendocrine. We haven't really focused on that today, but it was part of the data set. So I think in terms of the aims, as I said right at the outset, risk stratification remains a real challenge in prostate cancer, and high-risk patients, a lot of patients get put into this category of high risk. But within that, you can see that by the fraction that the recurrence rate is about 50-50. And so knowing which patients within that 50-50 are going to go on to recur is quite key to how you manage their treatment. And I think one of the things that I've really learned at my time at ICR is just how much cancer can be seen as a disease driven by evolution. So this is a real foundational paper in the field of cancer evolution by Peter Noble, Science 1976. And you can see this idea of you start with, your disease here, and at each stage, there's mutations and branching off. And so what you have is not necessarily a tumor that's all one thing, but rather it's heterogeneous. I mean, we see that in the morphology in the tissue, in IHC expression, but it's also happening at a genetic level, that you are having these branching events happening. And so there might be one subpopulation, which is the subpopulation that goes on to develop metastatic potential. And so being able to model this evolutionary behavior, and if you can rewind the clock all the way back to the point of diagnosis and say, well, this patient has the evolutionary potential to go on to develop metastasis. Well, that's a really powerful thing. And so I think that this was the one question we wanted to answer. Can methods of evolution give us that insight to really predict that trajectory that patients are on, beyond what we currently know within the existing risk stratification protocol? And I think on a more general level, I think we were quite interested in looking at sort of histological and genomic metrics that are predictive of patient outcome, and potentially metrics that feed into each other, that there is some sort of association we can find there between genomics and histology. And I think one of the very important aspects that I'd like to emphasise is that we were completely blinded to clinical outcomes. So we had to essentially decide from the outset what are the sort of things that we want to actually measure, and then compute all our metrics, and then open the box at the end, which comes with its challenges. And so I'm going to be mainly focusing on the imaging cohort, and we'll talk a little bit about the sort of cohort of sequencing. But as you can see, while there is a majority of cancer, samples, there is a certain proportion of normal, just where you missed, with the needle. And so, what we're going to be looking at today is predominantly from this cohort. So this is sort of in a slide, the image analysis where the digital pathology work, and some of this stuff will be quite familiar to you. And so essentially there are two sort of arms that I've looked at. There is the cell classification, which is built around an SCCNN style cell classifier, which is something that was pioneered here at Warwick. It's something that I've definitely benefited from working with people who innovated this very quite incredible sort of move forward in terms of digital pathology. And we use this to essentially identify three types of cells: epithelial cells, stromal cells, and immune cells, which was based on what we had access to in terms of annotations. And so this allows us to explore things like the epithelial stromal ratio, the presence of chills, and try and understand on a cellular level what's happening in the tissue. But of course, I think one of the foundational things within prostate cancer is this idea of Gleason grading. Now, what you see on the right here, these are some drawings by Donald Gleason. They date back to the 1960s. And the fact that we're still talking about Gleason grading today is really a testament to the enduring power of this grading system. Of course, it has changed over time. I mean, at this point, you have this scale of 1 to 5, going from very normal-looking glands to slightly more, aberrant glands, all the way to glandular structure disappearing completely. But today we look at it in terms of 3 grades, 3, 4, and 5, but still very much still going from having relatively normal-looking glands with slight aberrations, all the way to just fields of cells, essentially. And I think, we talk about heterogeneity a lot in cancer, and I think that very early on, Donald Gleason understood the importance of heterogeneity, because the grading system has this primary and secondary grade. What is the most common pattern we see in the tissue, and what's the second most common pattern? And you basically have that as a score. So if you see mostly 3 with a bit of 4, that would be 3 plus 4, which is sometimes reported as, you add them together and have a 7. And I mentioned that there had been a grading review back in the mid-2010s. And they then came up with this idea of a grade group system, which is what I'm going to mainly be working with today, because that is the clinical standard. And you can see here they're going from grade group 1 to 5. You get this very clear difference in terms of risk of recurrence. And I think one of the key things here was this idea of 3 plus 4 and 4 plus 3 being different, and adding them both and saying, okay, this is grade 7 is not sufficient. You need to break these two up. And so most of what I'm going to be talking about today will be using this grade group system, but behind the scenes it is up to the primary and secondary pattern. So that is how Gleason grading works, but how does it work in an automated setting? And normally I go into this in a very surface level, but as I'm in a group of people who do this on a daily basis, I'll go into a bit more detail than normal. But essentially, the Gleason deep learning. And so just on a very surface level, we go from having a whole site image to having these annotated regions, and we can identify benign glands, but also these pattern 3, 4, and 5. And so from this, obviously this is a bunch of segmentations, but we can create an estimate of what is the balance of 3 and 4, and then use that to convert it into primary and secondary grade, and use that to develop, to generate the sort of ISO, what you saw there, the grade group 1 to 5. And we'll talk about some of the specifics of that in a second, but I also wanted to use this as an opportunity to talk about the specifics of the model, because often, I don't get to talk to digital pathology people, and so I don't get to say, hey, look, my cool model. Well, I think it's pretty cool. But basically, it's a foundation sort of UNet sort of thing. So, I think that's, in terms of focus in deep learning, new models sort of come along since then, which arguably can't compete this. But I think that, at the time that I was developing this was sort of state of the art. And I think one of the key things here, and you can see this if you look at the training data, is that it's based on a sort of concept of multi-resolutionality. So it's not just a single image patch. You take a 10x and a 20x, and it uses that to sort of come up with an assessment for the Gleason grading regions. And We tested this with a few different combinations of resolutions, and basically there's definitely an improvement if you're from just using a 20x to adding next to a 10x resolution. Beyond that, you don't tend to see too much of an improvement. I think so we're sort of 20x and 10x there. And yeah, as I say, that this is sort of built on sort of ResNet layers on the sort of encoding layer. And you can see at the end you get these sort of masked regions, and it's essentially whichever one is that has the highest percentage per pixel is selected as the segmented region. And so yeah, that's basically a high level idea. Yes, guy. Oh yeah.
So how are you incorporating the sort of the previous from the architecture?
Oh, so
how did you incorporate the multi-res? If you go back to the architecture.
Yes. So you can see it. So if you look at these, so each of these boxes has been think about as like one layer. So you've got the key down here, and so you can see there's essentially two happening in parallel. And then you get this concatenation here, and then it feeds forward from there, basically.
So you're incorporating the 10x one block down.
So no, so you can see like you have it. So essentially you start here. So it's kind of hard to see here, but this is the 20x. Then behind you've got the 10x, and they both feed into sort of like a parallel encoding through each of the steps, and then it gets concatenated, and that feeds into the decoding essentially. Yeah.
So essentially, you have a 6-channel input. Is that right?
Yes, that's true. Yeah. But it's important to emphasize that, like, in terms of like I suppose ultimately you could think of it, you could probably express the convolution in terms of a 6-channel at end time, but it's actually sort of two separate sets of parallel layers.
So basically 2 units in parallel.
Essentially, yeah.
One at 20x, another one at 10x.
Essentially, yeah. Okay. And then there's a concatenation. So as I say, I think that there's definitely been innovations in the recent years that would perhaps be up for what we did here. But I think that as I say, it was able to give us some good segmentation accuracy as we'll see in a second. And so I think one of the things I like to say is that this is sort of the raw information. And from this, we wanted to sort of identify the tissue features that were associated with disease evolution to be able to make these predictions, sort of where's the trajectory of the cancer, and ideally be predictive of outcome beyond that. And as I said, we were blinded to outcome at the beginning, and so it was a little bit of a nerve-wracking moment where we had to really choose wisely what are the things, what theoretically are the things that we think will be most important here. And pick those and really turn that at the beginning, because once you open the box, you can't go back, you can't rewind again. And so it was key that we do our best to get that right at the start. So yeah, what sort of things do we do to get that information? And I want to take you back in time to this was the 28th of June 2021 apparently, and we were in a room not entirely dissimilar to this one, and we were sort of like a board, and we just sort of wrote out all the different things we thought would be important. So you can see on the targeted sequencing, you have all these mutations within your panel. You can look at structurality. In the low-pass whole genome, we have a whole bunch of different ways of expressing essentially the heterogeneity and the evolutionary potential of the tissue, along with things such as ploidy and PGA. And on the image analysis side of things, of course, we have the Gleason score, so we can look at high versus low, different grade groups, but also this idea of heterogeneity, which we'll revisit in a second. And on the cell classification, I mean, a lot to do with sort of infiltration, immune infiltration, and lots of different ways of expressing the amount of immune potential within the tissue. And so just sort of clean that up for a moment, these are sort of the pathology metrics that I mainly looked at. And so we'll start out looking at the cell classification. So I came up with, I sort of adapted a few different ways of looking at the amount of immune cells within the tissue, with the cell percentage, like amount of clustering, which is based on something called the Irani score, which is common within it's not necessarily a cancer, but like prostate inflammation score, and as well as ITLR and molecular feature index, which were innovated within one group at the ICR, and so seem like good ones to do, as they've been shown to be very predictive within other tissue types. Unfortunately, we didn't really see anything, and I think I quite like to present sort of negative results from time to time, because I think it does it, particularly in this sort of setting where we had to sort of set a selection of features right at the beginning and say, okay, these are the things we think are important, and sometimes you will miss, and you will find that there isn't really anything. But there is actually some good news here that in recent work we've been looking at. Yes.
You can finish your sentence.
I was going to say, in recent work, there may be a sort of a happy ending to this side of things, but it was a bit of a disappointment at the time when we were blinded that, yeah, there was nothing really happening in the immune activity.
So if you were blinded to the outcome, how did you compute these results at the beginning?
Well, we were blinded to the outcome data, so we didn't know like time to metastasis, time to recurrence, but we had access to the the histopathology slide, and
Right.
so we could detect cells, we could do Gleason grading based on the
But to
pathology.
do this, you need the outcome data, right?
This would have to be unblinded. So like this was
Oh.
when we unblinded. I did these.
So
Kaplan-Meier.
you send the features and they will compute these Kaplan-Meier curves.
Yes. Well, basically we had a clinical PhD student who collected all the data, and so we out to her, and that was how we these metrics. And
So she would compute these, but she wouldn't share the clinical outcome data with you.
well, we now know the clinical outcome data, but that was
Right.
that was
But at that
point,
time,
yeah. So
we're talking about when you were designing the whole thing.
yeah, well, when we were designing the whole thing, we didn't have access to these Kaplan-Meiers, we didn't have access to anything. We just had to decide, okay, these are the features we're going to be exploring.
Also, you didn't even know that these results were going to be negative.
Yeah,
Okay.
that's why I was saying it was a bit of a daunting experience. I think with hindsight, perhaps there'll be things that we would do differently. But yeah, we set about, okay, these are the things, and obviously Catherine, who was the clinical PhD student, she didn't know that they would be negative either, because she had the clinical data, but she didn't have access to all the metrics, and so it was only at the end when we were blinded, we put this all together and start to see what's significant. And yeah, I think that's a lesson. Sometimes you get negative results, and that's not necessarily a bad thing, because sometimes the answer is no, and that gives you information in and of itself. The good news was that things were a bit better on the Gleason grading side of things. So yes, no, sorry.
As we're alluding to here, Gleason grading is typically used for understanding how patients are going to do down the line. Did you have any intuition that the immune markers would be potentially useful?
This was based on, well, first off, TILs are often a key indicator of risk within a number of different cancer types, and there have been some studies which suggested that, unlike with a lot of cancer types where it is a positive thing, high TILs is generally positive for outcome. The opposite has generally been observed within prostate cancer, but it's a bit of a mixed picture. And so I think that we chose that. Well, I chose those as features because it was like, well, this is an opportunity to really shine a light on that. And unfortunately, we shined a light on it, and we didn't really see a whole lot. But that was the intention behind it. That TILs are complex within prostate cancer here. And as I say, we'll see a bit at the end about how maybe we can regain some clarity on that issue. But yeah, so in terms of Gleason grading, we had 3 types of grading. So as I say, there was back when the patient was first diagnosed, we had their original pathology report, so the standard score we got from them. And we also had the central review, which happened in 2016, with the modern scoring system. And both those were a standard score, so like a 3+4 primary, secondary pattern. Whereas in the case of the automated classifier, we had a detailed segmentation like what you see on the right. And so from the sort of detailed segmentation, in order to have a like-for-like comparison, we convert to this segmentation mapping to a primary and secondary grade, which is essentially based on the amount of each pattern. So we basically add up all the pixels and see which ones are the most common and second most common. Which potentially there are as I've learned, sort of subtleties with the way that pathologists handle it. That perhaps if you have more 3s, let's say you have an equal balance, not an equal balance, but some 3, some 4, and some 5. The fact that there's a presence of 5 might make you automatically say that's your secondary pattern, even if there's more 3 technically. It's it is, there are sort of subtleties, but that was the way that we formed this. We basically counted all the pixels and used that to develop the primary and secondary pattern. And so what we wanted to see was, okay, we've got these 3 sets of scores. How do these compare in terms of predicting time to recurrence? And again, this was all using the grade group system. So while we had a 3+4 in each of the categories, we converted this to a grade group. And one of the things that, again, the first piece of good news following on blinding was that there was apparently a much better prediction in terms of the automated classifier as compared to the original under this scoring pathologist. And I think that the original pathologist, I think there was certainly data collection issues in the sense that the way in which Gleason grading had been performed varied across time. But the fact that we still saw this within the reviewing pathologist, where there was a bit more separation, but you can see the separation isn't necessarily tied to the order of the groups. You can see that group 5 is actually higher risk than group 4, according to the Kaplan-Meier here, in terms of prediction of recurrence. Whereas you can see here that, particularly the separation between group 4 and group 5, there is a pretty substantial split. And so this was, something that was very, exciting to see. And yeah, like group 5, you have like 50% patients recur within 100 months. And so one thing we want to know is, well, what's happening behind the scenes? Why is it that the digital, because it's nice to say, okay, the classifier is better, but also when you think about it, the classifier was trained on human annotations from a pathologist. So how is it outperforming a pathologist at the end of the day?
A comment here from Mustafa said, related to TILs and prostate cancer, I've also found in my research that high or low expression of T cell CD8 is not associated with good or bad outcome, progression-free interval.
Oh, interesting.
Whereas patients could have been stratified by the mitotic activity. So a comment's not a question, but
I mean, I think it is a complex picture. I think that you see studies that say different things. It might be that the subpopulation of immune cells that you're looking at, matters there. It's and I've seen some things about like interferon signaling being important, for instance. And yeah, as I say, I think there's also other things at play which I'll talk about at the end. But as I say, that was sort of where we stood.
I think it's basically most of us saying use his mitotic profiler rather than TILs or anything else.
Oh, fair enough. Well, yeah, I'm happy to give that a go. But as I say, in terms of like Gleason grading, I think one thing immediately stood out in terms of the difference between what the classifier was saying versus what the pathologist was saying, and it really comes down to these big categories here. That there is this big population of Group 5 according to the pathologist that was coming out as Group 4 according to the classifier. And so we wanted to sort of explore why that's happening. And essentially, I came to the conclusion that when we came to like taking our sort of segmentation and converting it into a primary and secondary pattern, and then going from because we have multiple samples per patient is nothing to emphasize, so aggregate multiple samples into a single patient score, we had a very strict definition of what should be Group 5. Essentially, every individual slide should be Group 5 in order to qualify as Group 5 according to the automated classification, which is not necessarily the way the pathologists do it. In fact, it would probably be the inverse. If you had a single Group 5 sample, the pathologist would automatically boost you up to Group 5. So I think the first thing to say is that the pathologists are doing nothing wrong. They're following existing scoring standards, and that's perfectly acceptable. I have no question about that. But nevertheless, in this situation, the automated classifier was outperforming the pathologist, and so the question is like, how do we reconcile this? How do we come up with some sort of answer to, like, okay, the automated classifier is outperforming by doing the wrong thing? And I think what I concluded here was that perhaps, because these are high-risk patients, you have to remember, that most of them will have a high Gleason grade, a very, very large number go straight into Group 5, but maybe Group 5 is too all-encompassing. It might work for a general population where you have a very small proportion of these sort of patients, but when you are looking at these patients in isolation, it's not granular enough. But there is a high-risk group within a high-risk group, and so perhaps Gleason grading needs to be refined to reflect this. That there is a further feathering that could allow us to predict that okay, these patients are high risk. We need to pay more attention to them. We need to escalate treatment, for instance. And I mean, I think there's still questions to be answered there as to are there options in terms of escalating treatment, or is it just an indicator of outcome? But I think this is quite an interesting case because when we look at digital pathology, at least pathology, you often sort of see two main arms. You have this idea of using tools within the clinic that taking an existing scoring system such as Gleason grading and making an automated system that can be rolled out in the clinic to essentially emulate what pathologists are doing. And then you have more exploratory analysis where it's looking for novel biomarkers and trying to identify those. And I think that this sort of shows another way in which Gleason grading in which sort of digital pathology can be applied, in which it can show how a pathologist can do potentially a better job or work better with specific patient cohorts. And I think that that's an area that I'm quite keen to sort of explore further. How can we use AI to not just make better tools, but also to make pathologists who can give better prediction of outcome, really. So that is a sort of standard grading, but I want to now move on to sort of another way of looking at Gleason grading, because I think if we just simply take the segmentation and convert down to a primary secondary pattern, we're really underselling what we can do with this this sort of technology. And I think it's not to say that standard Gleason grading is a problem, but I think that it's low resolution by necessity. A pathologist can't segment out all the readings, it's just not feasible. But if we do have this additional information, does the distribution of primary and secondary gland tell us anything additional? Is there a difference between sort of a homogeneous pattern and a heterogeneous pattern, as you can see here? So you can see, for instance, here with the 4 and the 5, there is very much a blockiness, that there is like transitions between the two, but they are largely sort of homogeneous areas, whereas you can see for the heterogeneous, there's this marbling pattern between different areas that changes quite a lot. And so the question is there a difference there? Are they just different ways in which is a 3+4 that is blocky versus the 3+4 that's all mixed together? Are they different, or is it just, they're both 3+4s and they just look slightly different, basically? And one thing I wanted to do was try and capture that level. So I looked at it in terms of the Morris-Eater index, it's a measure from ecology, basically. So it looks at whether populations are sort of segregated or are they mixed together. And you can also use this to look at the mixing between different cells. And so again, the Indian one used this to look at the mixing between immune cells and epithelial cells. But you can also look at it in terms of like glandular cells that are part of the pattern 3 region, and glandular cells that are part of the pattern 4 region. And obviously, if you have these blocky regions, you should see a lower Morris-Eater index, you should see more of this. Whereas if you have a very high Morris-Eater index, that suggests you've got more mixing. And so, yeah, essentially it was a case of combining the 2 bits of information. We have the cell classification, so we strip out all of the stroma and immune cells. We then take the epithelial cells, map it onto the segmentation mask we see at the bottom, and use that to get a combined sort of cell distribution of epithelial cells of different Gleason patterns. And so the idea is that then, does this tell us anything additional beyond the standard Gleason grading? And so, yeah, you can see this is sort of the examples from before. You can see this one has a very low Gleason Morris-Eater index, and this one has a very high Gleason Morris-Eater index. And so I was quite pleased to see that, again, this was a predictor of outcome. And so the patients who have greater heterogeneity appear to have a greater likelihood of recurrence. And this held up also at the multivariate level. And we were sort of trying to understand what this might be, the sort of meaning behind the, under the hood. And I think it all comes down to this idea of evolvability. What is the likelihood that this sort of pattern that you see here is going to stay that way or is going to change? And so my feeling is that if you have these sort of large homogeneous regions that may be separated slightly, these are things that have already reached a sort of evolutionary stable state. And while they may go on to progress, they're less likely to than something where you are perhaps capturing this transition.
Sorry, just a couple of comments on that.
Okay, sure. If there is any comments, and so just to say, this was also held on, so we may look at time to recurrence in this presentation, but also these things held up in terms of metastasis. So patients who have this level of mixing also are more likely to develop metastasis down the line. And so I now want to jump over quickly to the sequencing cohort. So again, this is a subset of 111 patients. They are so again, 568 single regions. We also have some where we have multi-regions on a slide, which you can see have been annotated here. So generally, we would take like a whole slide, but if you have lots of cores on a particular slide, then we may split it up into multiple sequencing runs. And so this led to, like I say, 568 single regions plus 35 multi-regions, each with a single sequencing run. And so, yeah, as I say, I think that one of the benefits of this project is that we have this matched niche, so we can now sort of say, okay, well, how does this combine with the, how does this sequencing information combine with the digital pathology? And so we looked at it in a number of different ways, and I'm going to focus on one. So I've mentioned a lot about this idea of diversity, of evolvability, of heterogeneity, and so can we see sort of like a combined heterogeneity within the tissue? And I think, again, this happened sort of independently, so we unblinded, and so George, who again, I did not do any of the sort of bioinformatic sort of copy number analysis or anything like that. So that was all his work. Excellent, bioinformatics researcher, excellent, copy number guy, as he likes to call himself. And I think one of the things that I was going to say, we always like to make jokes about it. But yeah, like I say, excellent. He's now a PI over in Austria, so yeah, doing very well for himself. And I think that one of the things we saw immediately, so this is from the panel sequencing, as I said earlier, you can see these are the sort of panel of the main sort of prostate cancer driver genes. And I think one of the things that would really come from it isn't really a dominant sort of, in some cancer types you see, like for instance, TP53 just dominates everything, it's all over the place. Whereas here there isn't really a single dominant one. In fact, for about half of the patients, there's no real sort of mutation in the panel that jumps out at all. And you can see, so you see here this clonal and subclonal. So anything that has a lighter pattern here, these are ones that are present in some of the patient samples, but not all of them. So you can see there is a great amount of heterogeneity, even within a patient, even within a prostate, with like different regions within the tumor have different mutation combinations. I think that this notion of heterogeneity also when we moved over to the copy number analysis, the low-pass whole genome sequencing, you can see that like while there's a certain number of mutations across the genome, so these are all the different genes, if that's in sort of lots before, so chromosome 1 all the way through to 22 and X and Y, you can see that like there's a certain proportion that are shared. These are where a certain proportion of the patient's samples contain a gain or a loss of a particular chromosome or chromosome arm. But there's a fairly substantial proportion that are unique, that only one sample within the patient has this particular gain or loss. And so we now wanted to sort of, well, this sort of led to this idea of, well, if patients have lots of heterogeneity across their samples, so they have lots of these sort of gains or losses that are exclusive to one sample, is that indicative of outcome? And you can see that George developed this idea of the Spearman correlation. So essentially developing sort of a copy number profile, like all the copy number profiles for that patient, are they mostly correlated? Are the gains and losses all in the same place, or are they in different places? And so the more correlated they are, the more homogeneous you are, the less correlated they are, the more heterogeneous that you are. And one thing we saw is, once again, that like more heterogeneity, and now we're looking at sort of genomic heterogeneity within your copy number profiles, is again a valid indicator of outcome. And it was really, really encouraging to see that both from an imaging perspective and a genomic perspective, like after unblinding, both were pointing in the same direction, that they were both showing this idea that evolvability of greater heterogeneity was in fact predictive of increased risk of recurrence within prostate cancer, and these high-risk prostate cancers. And so that led to the question, well, this is individually, but what happens when we put this together? Are these things showing us the same thing? Are they showing us different things, like things from different angles? And so that was where we started to move next, this idea of joint diversity, combining this Gleason morphology from the digital pathology with Spearman correlation from the copy number analysis. And so one thing we found was this appears to be more than a sum of its parts, that it was a better predictor than each individual metric, and really when you put them together, things really start to pop. So you can see here, this is the Kaplan-Meier. So once again, this is like this red line here are the patients who are basically double high risk. They are high risk according to Spearman, they're high risk according to Gleason morphology. And one thing we found is there's no correlation between the two. And so it appears that they're showing us different things, different kinds of heterogeneity with the samples. And I think, perhaps different resolution. So for instance, the Gleason morphology, you're looking at within sample heterogeneity, local regions within a single core, whereas the Spearman is heterogeneity across samples, like, the region over here having different mutations to a region over here. And it's really when you are double heterogeneous, you're heterogeneous on multiple scales, that's when you have the high risk. And what was particularly encouraging is when we split it out into, high, low, low, high, low, low, and high, high, it's really only the double high risk patients who have any difference in terms of outcome. If you're high risk according to one, the low risk according to the other, your outcome is the same as if you're low risk according to both. So it is definitely, as far as this analysis seems to show, this double heterogeneous, this like different resolutions of heterogeneity and evolvability that really is the predictor in terms of higher risk of recurrence in prostate cancer. And I should mention, while I'm showing here recurrence, we also see the same thing in terms of risk of metastasis and risk of going on to develop castrate resistance and require a different second-line therapy. And again, this is something we saw and also holds up in multivariate analysis. And so just to give some basic conclusions, I think that definitely, as I showed earlier, there's definitely ways to potentially augment traditional Gleason grading to provide better prediction for patients of high risk. I think this further stratification of group 5, I think, is something that I'm quite interested to pursue further. But I think also, like, looking at not just primary and secondary, but more encompassing description of heterogeneity. As I say, heterogeneity has been a cornerstone of the Gleason grading system, but being able to go another level may be able to give us an additional factor that allows us to predict risk. But I think also this whole idea of using evolutionary metrics to be able to predict right at the point of diagnosis which patients are more likely to go on to develop bad outcomes. So this go from having a 50-50 risk to be able to say more about, okay, these are the patients who are most likely to develop a recurrence and go on to develop metastasis. It appears that that's a promising direction that should be pursued further. And I think that particularly it's strongest when evolution is measured at different scales and from different angles. That I think that this has been one of the things that I think has been really rewarding about being in the group I've been in at ICR, is that having all these different disciplines together has allowed us to sort of see the cancer from different angles and give us this additional strength in prediction. And of course, I think it's important to mention that this is a retrospective study on one particular clinical trial with one particular risk like level. And it's important that these findings need to be validated further in external data sets to really understand, how these findings apply elsewhere in terms of risk. And that's something that I'm keen on pursuing, moving forward. And so, yeah, I just wanted to quickly give my thanks to some of the main key players within this project. So Javier Fernandez, he was the one who prepared all the samples. As I say, it was really not easy to get good quality copy number data out of these FFPE samples. And he spent a very long time in the lab refining things through many, many rounds of reworking things. And it's really his credit that we, got to where we did in the end. And certainly, in terms of the analysis of the copy number data, George has been, instrumental in that. And he's been a great person to work alongside. And I've learned a lot about genomics just in that process. And certainly, on the clinical side, Kat, as I said earlier, she was one who collected all of the spiral data. And I mean, she'd always be sitting, when we'd be talking about ideas, and what should we be looking for? She'd be sitting there in the corner on her laptop, knowing all the answers, and very good at not revealing anything. But yeah, she was, in terms of also talking about this idea of, okay, what's clinically relevant? Are the things we're suggesting making sense in terms of how clinical practices today should, excellent voice. And of course, Andrea and David, the two sort of PIs on the project, both from the sort of scientific but also the clinical side, and both have been great at sort of allowing us to share this work and think about where we go next and everything. And but definitely have been it's been rewarding to work with both. And there's a whole other set of people who have been incredibly helpful. Kathy Corbishley, the pathologist who did all the regrading, helped me with training of the Lucy Redding classifier, Christine Stossel, who did a lot of the collection of the FFP blocks across many different centres, and Basilis Stavridis, who I've been working with a bit more closely in recent months, has helped me in collecting all the data for the cell classifier. And so yeah, across the funders as well. It's been an excellent project, and I haven't traded it for anything. It's been a really amazing time. And I realize I'm coming up to time, but if I'm allowed a few more minutes, I just want to talk about where I might be going next. And I'm not going to go into too much detail, but just to give you a bit of a flavour of what might be coming next. So again, Catherine, one of her PhD projects has been this idea of testosterone recovery. So I mentioned previously that patients are given androgen deprivation therapy, and they're given it for 1 to 3 years. And after they come off, their testosterone levels will start to rise. Sometimes patients going to recover their testosterone due to age and potentially due to the effects of treatment, causing long-term tissue damage. But you can see that to say 10 nanomoles per litre, which is the low end of normal, you have around sort of 50% to 60% at that level. But this isn't necessarily, obviously it's a good thing because long-term, testosterone expression in general comes with a whole host of side effects, both in terms of quality of life and also risk of other comorbidities such as heart conditions, for instance. But it also increases your risk of cancer. So the patients who actually recover their testosterone are more likely to recur, they're more likely to develop metastases. And so I thought this was an excellent resource, and you can see here that we have a very good overlap with our IMRT analysis for the wider forecast project. And so one thing I want to know is, well, how do these evolutionary metrics play with, because of course we're looking at diagnosis, but there's a lot happening in the intervening time, particularly like testosterone levels rising. How do these testosterone levels, fit in with everything else? And this is just one sort of couple of lines to work your appetite there. The paper hopefully will be coming out, I'm hoping to get it submitted by the end of the year, so hopefully it'll be out sometime next year. But you can see that if we split out these lines, so this is the high risk according to the Spearman's row, this is the low risk, that when we split out in terms of recovered and didn't recover, it's only the patients who didn't recover their testosterone who have the true low risk. The patients who are predicted as low risk but do recover their testosterone are the same as the patients who are predicted as high risk. And so one of the unfortunate conclusions there is that you're only low risk as long as your testosterone is suppressed, and that has implications for how we potentially might treat ADT, moving forward. And I think that there are some silver linings there. Maybe it's not necessarily suppression to zero that might be useful for preventing this sort of thing. Maybe there's a different level, but this is something we're still investigating. And one more I want to look at, which again I said about negative results earlier, the immune cell percentage or infiltration in general didn't really show much. But when we look at it in conjunction with testosterone recovery, we start to see something quite interesting. That the patients who have low, immune column, they don't have much inflammation, immune activity at diagnosis, there isn't really a difference between the patients who do and don't recover the testosterone. But the patients who have like a lot of inflammation at diagnosis, there are quite substantial differences between their, the patients who do and don't recover their testosterone. Patients who do recover their testosterone have very bad outcomes, whereas patients who don't recover their testosterone have, markedly improved outcomes. And so, I think this is an interesting sort of suggestion that maybe there's some interplay between immune activity and also the, and this is sort of based on immune activity at diagnosis versus the sort of potential long-term effects of androgen deprivation therapy in terms of its ability to suppress the tumour. And so that's just a little bit to whet the appetite, but that's still where I'm going next. And I've gotten quite keen into looking into the effects of testosterone recovery on this one. So yeah, hopefully there'll be something out on that in the new year.
Well, thank you very much. Fantastic talk. Before we go on to the questions, I know it's 3 o'clock, so if anyone does need to go, please feel free to run out. So we've got a couple of questions online, but has anyone got any question in person to start? Yeah, Simon?
So with editing the polycell rate, this merits instead of using more using the segmentation of the bands and then using machine generated features.
I suppose ultimately, I think that those machine generated features are, I think there's, I suppose ultimately, you are creating a set of criteria one way or the other. And I think that, there is variability on sort of the way you train a model versus variability on a human assessment. And I think that, I suppose the question is, where is that variability coming from? Is it purely human assessment? Is it interpretation of the definitions? Or is it that, there are specific subtypes where it is difficult to draw the line? And I think that in that case, it's difficult to draw the line in terms of having those features, because you can be more objective about it. It's not just this looks kind of 3-ish. It is 3-ish because of these particular criteria. And so I think it's really finding the window of opportunity there. But I do think there is definitely having more detailed criteria, because it goes back to my point about this idea of making just better potentially by using like AI. Maybe if we have like these robust criteria, it can be used not just within a machine learning model, but also can be used by humans as well.
We have a couple of questions online. So Yuri's asked a couple of questions. So he said, why don't you include TP53 BRCA status into your models? Also, FOXA1 and SPOP mutations were associated with response to ADT and might significantly affect the prognosis in your particular clinical setting. It might be easier. Might make that a bit bigger so you can also see that.
So I think there's a few things to mention. So TP53 is an interesting one. I don't have the in one of my slides, but there is an association between TP53 status and Gleason grade within our cohort. So I think that that is something that is there is definitely merit to looking into that. But one thing I would emphasise, and it goes back to what I was showing previously, if I was to jump back to the sort of the panel sequencing, I think that one of the things that really jumped out to us is there isn't really any dominant mutations there. I mean, there is TP53, but you're talking about a very small number of patients, and a very even smaller number that it's actually prone, or so it's. It's not saying that it isn't a valuable like component, but I think it's one of those that if you're looking for sort of more like you want to sort of say, okay, 50-50 patients which 50% are going to go on to recur. It's a low. I think it would need to be part of a much larger sort of set of criteria. As I said, the fact that TP53 status does correlate with Gleason grading is already an indication that it's something that is worth paying attention to. But I think that it's again, it's I suppose probably more comprehensive collection of criteria would be needed.
Brilliant.